Okay, it is started. All right. So I should be on my whiteboard now. So obviously when I have my whiteboard, I can't see anything that is happening on the other side. So if there's something you don't understand, you need clarity on, or you have a question, please do let me know so that I can attend to your question. Just unmute and ask your question and just say, ma'am, so that I can respond. So let's look at mitosis first. So if you were to be asked to define what mitosis is, you would need to say that is a type of cell division. That is a type of cell division. Please do listen to what I'm saying, just in case the pen doesn't write properly, as it's doing now. Cell division. This is a type of cell division whereby one cell is going to divide to form two cells. It's going to divide to form two cells. These two cells are going to be identical to each other. That word is important. These two cells are going to be identical to each other, meaning they are going to be the same. So at the end of mitosis, we form two cells, and these cells are identical to each other and to the parent cell. They are identical. As soon as you join Teams, the pen is lagging and it becomes slow. Apparently, Teams takes up a lot of space when you are running a meeting. So these two cells then become identical. Now, the question would be, how exactly will they become identical? So if a cell has 4 chromosomes, of which I'm going to speak about, if a cell has 4 chromosomes, this is the parent cell. That's the cell that is going to divide. When this cell divides, it will form two cells. Now, each of the two cells will also have 4 chromosomes. Now, this does not make sense mathematically because you would think, okay, if one cell divides to form two cells, that means we are going to halve the number of chromosomes, and each cell will end up having half the number of chromosomes. But that is not the case. We then say in mitosis, the chromosome number is maintained. How is this possible? Remember, in our second lesson, we spoke about DNA replication, and we said this DNA replication occurs in the nucleus of a cell, whereby DNA is going to replicate and make a copy of itself. So this is exactly why DNA replication has to take place before cell division. So when this DNA replicates to form a copy of itself, when the cell divides, the cytoplasm, which is the jelly-like substance, is going to split over here to ensure that we are having two cells. This will become one cell, and this will become another cell. So now we had a copy of DNA. We ended up having two DNAs in one cell. When the cell divided, we ended up having two cells and each cell with that DNA. Now we remind ourselves of what I've drawn here, which is the chromatin network. These lines that I've drawn here, they will condense to form what we call chromosomes. So these lines, which is your chromatin network, during cell division will condense, become closely packed to form what we call chromosomes. Now, these chromosomes carry our genetic material, they carry the DNA. All of this background is very important. The chromosomes carry our genetic material, our DNA. So this same DNA that I was speaking about here is the same DNA that will be carried by chromosomes. Now, me and you as human beings, we have 46 of these chromosomes. And each chromosome will contain a DNA that determines a certain characteristics for an individual. So there'll be certain genes, sections of DNA in the chromosomes that will code for a characteristic, meaning this part may be of a chromosome over here, will code for your hair color, hence your hair is brown. Another part of a chromosome will code for your height, hence you are short or tall. Another part of a chromosome will code for your skin tone, hence you are light skin, dark skin, brown, or whatever. Now, the reason you have all of these physical characteristics, they're actually determined by the DNA that we find in the chromosomes. Now, what I've drawn is one chromosome. So we call it a chromosome as a whole there. That is called a chromosome. I am about to confuse you. So please make a note. Please come to the party and just write. Okay. All right. So this is a chromosome. It's detecting my hand. Don't detect my hand. Okay. So this is a chromosome. Just please do write it on your side. I'm fixing my pen. Chromosome. Chromosome. It's M-E at the end. Now, maybe, okay, I have to do this now, not later. So what you're seeing on your slide is a chromosome. Now, the question would be, what are these two threads that you are able to see? So a chromosome is made up of a chromatid. Two words here. A chromosome is made up of a chromatid, but if we're to look at this particular chromosome, this is a chromatid and this is a chromatid. So what you have here is a chromatid. Chromatid is CHROMATID. That is a chromatid. The same thing over here is also called a chromatid. That means a full chromosome is made up of two chromatids. So when these chromatids are joined together, so there's a dot in the middle that I've indicated here. That dot in the middle is for what we call a centromere. The terminology will probably give us a headache today, but that's how the topic is. Centromere, this is M-E-R-E at the end, M-E-R-E. So what exactly do we have here? A chromosome. A chromosome, in our example, is made up of two chromatids. Now, these chromatids are joined together by what we call a centromere. But we can also have something like this, where we are just having this thread over here. So I want us to compare the two. This thread over here, and I'm going to put a dot, and these two strands over here. Now I want us to listen. This strand over here is called a chromosome. This one. We call it a chromosome. This one over here is also called a chromosome. Now the question would be, how are we giving it the same name while they look different? Please make notes everywhere. Whenever it comes to these diagrams, they can be confusing, but if we get them now, we should be fine. This is a chromosome and this is another chromosome. Let's understand. This is a chromosome that is made up of one chromatid. So this is a chromosome that is made up of one chromatid, that thread, only one, and that is the chromosome. Now, in other textbooks, you might find this one chromatid or one chromosome made up of one chromatid, and then it will have a dot in the middle. That will still be correct. That is just indicating the centromere. Now, during DNA replication, this very same chromosome made up of one chromatid is going to replicate, make a copy of itself. So once this one chromatid makes a copy of itself, meaning this is the chromatid, we need a copy of it. There, during DNA replication, the same DNA replication that we did, it will make another chromatid. And then we end up having this chromosome also. But in this case, this chromosome is made up of 2 chromatids. Chromatids. That is the only difference. So this one will eventually replicate and form another chromosome that is very, very identical to it. So what we had initially was a chromosome made up of 1 chromatid. Then in this diagram, we had a chromosome made up of 2 chromatids that are joined together by a centromere. Now, when this chromosome is only having 1 chromatid in this state, where it has only 1 chromatid, we refer to it also as an unreplicated chromosome. You have to add all of these terminology. So just 1 chromatid of a chromosome, you can refer to it as unreplicated chromosome. This means that we are able to see a chromosome that has not undergone DNA replication. That is why it's called unreplicated. It's not writing. Yeah, there you go. Unreplicated chromosome. Please do listen to what I'm saying, just in case the pen fails. Unreplicated chromosome. When you are having a chromosome with 2 threads, you can refer to it with 2 chromatids. You can refer to it as a replicated chromosome. Replicated. What does this tell us? This tells us that DNA replication has occurred, and that means this chromatid has made a copy of itself. Unmute anytime and ask me a question, please, because this can be confusing. Okay, let me try and erase. Give me a second. I am erasing so that I can start asking questions. The intro is very important. We can't go to the phases without understanding the intro, because we'll have to come back to it. In each phase, we are going to speak about certain chromosomes, certain chromatids. There will be terminology that we include: daughter chromosomes, homologous chromosomes, and all of that. So I'm going to ask you guys, I'm going to ask you a question now. Just give me a second. All right. Can anyone tell me how many chromosomes are on the screen? Anyone? Can you tell me how many chromosomes you can see on the screen?
4.
4 chromosomes.
4.
That is correct. Okay. Then I'm going to do this. Okay. How many chromosomes do you now see on the screen?
8.
How many
8.
chromosomes do you now see on the screen?
Or 4.
Still 4. Still 4.
It's still 4 chromosomes.
4.
Yes. Okay. So I did say that the terminology will be a bit confusing. These are still 4 chromosomes. So the ones that we had initially were chromosomes that were made up of 1 chromatid, and they were unreplicated. So these were 4 unreplicated chromosomes, meaning they're made up of 1 chromatid. Also over here, these are 4 chromosomes. This is chromosome 1, chromosome 2, chromosome 3, chromosome 4. Now in this form, the chromosomes are replicated. And I think that is clear. Okay, we then go to another part. These chromosomes can also be referred to, these ones over here. So I think the first one I said you can refer to them as unreplicated. You can also refer to them as daughter chromosomes, but at a particular phase of mitosis and meiosis, which we'll get to. Now, these are just chromosomes, and we're going to leave it at that, chromosomes. Now, another one that we need to speak of is homologous pair of chromosomes. And the key word there is pair. When we speak about homologous pair, this is going to be long. It's going to take forever for the pen to write it. Let's see. Homologous, gas is G-O-U-S, homologous pair, normal pair, like your shoes or a pair of jeans, chromosomes. 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 Now, the key word here is pair, but I want us to remember something. When I spoke about chromosomes, I said me and you, if we're to zoom into one of our cells, we are going to get 46 chromosomes. Excuse me. And I said that in terms of your DNA, you inherited 50% of your DNA from your mother and 50% of your DNA from your father. The key word inherited, not received or found. So in life sciences, we're going to penalize you for that. I might make mistakes while I'm doing the presentation, but you have to use the word inherited because they were passed down to you through the gametes. But you don't say received or found. Okay, so with these 46 chromosomes that you have, this is your cell, me and you. Now that means if we're saying 50% of the chromosomes are inherited from each parent, we are going to half the 46. So the half of 46 is 23. So that means from the 46 chromosomes that you have, you inherited, this should be 23. It looks like a 28 now. Okay, so this should be 23 chromosomes. Okay, come on, come on, come on. 23 chromosomes you inherited from the mother, and another 23 chromosomes you inherited from the father. That's it. You don't inherit more from the other parent. It's not possible. Even if you really look like your mother or you really look like your father, you still inherited 50% from each parent. Now we'll get a look at that when we do genetics. Now, what you need to know about the 23 pairs of chromosomes. So if someone were to ask you how many chromosomes do you have, you'll say 23 pairs because that means 46, or you just say 46 chromosomes. Now, what you need to know about these chromosomes, the 23 from the mother, 23 from the father, they arrange themselves in pairs. So chromosome number 1 from the father will align with chromosome number 1 from the mother to form a pair. So that means in total, you're going to have 23 pairs of chromosomes. So 23 pairs still is actually 46 chromosomes. So what do I mean by this? So if we are looking at pair number 1, since we are having 23 pairs, so this could be the chromosome from the father, and then this could be the chromosome from the mother. And we're going to find them in the same position, and they make up a pair. Now, these two chromosomes, one from the father and one from the mother, we refer to them as homologous pair of chromosomes. We don't look at 46 chromosomes and say the first two are a pair. No, no, no. We only call them a pair when one is from the mother, one is from the father. And it will not be just any pair. There's certain things that will need to be the same in order for us to call them a homologous pair. Please note these three things. In order for us to call them a pair, they will need to have the same height. Homologous chromosomes will have the same height. Please make notes on your side because my pen is just lagging way too much. So it will be the same height. So that means this chromosome from the mother and this chromosome from the father will need to be the same height. They also need to have the centromere in the same position. The things that will need for them to be the same. It's like if you're having two shoes, you're going to call them a pair because they are both size 6, they are the same design, and all of those things. This is the pair of shoes that I'm wearing today. It's your Nike sneakers. They are the same size, same color, same shape, all of that business. Okay. So the first thing I said that homologous pairs of chromosomes will need to be the same height. And then the second thing, they will need to have the centromere in the same position. There is the centromere here. You should also have another centromere there. So the second point will be about the centromere. Please note that it will need to be in the same position. Now, they also need to have the same genes that they are coding for. Now, a gene, I've touched on it again, is a section of DNA. So if we are looking at this chromosome, in this section of DNA, we are going to have a sequence of bases, TAC, whatever. That section is basically a sequence of bases that determine your hair color. That is a gene, a section in a chromosome that codes for a characteristic. In this case, a characteristic can be your eye color, your hair color, skin tone, height, or whatever. So that means if we are looking at our chromosomes over here, what I mean by they have to have the same gene in the same position. This means that if the section of the chromosome from the mother codes for hair color, that means this section of the chromosome from the father codes for hair color also. So the third point would be speaking about the gene. Maybe there's another one, maybe I'm forgetting it. Maybe it's length, same height or length. I think they substitute those two words. Same size, yes, they have to be the same size also. Please do add that. Now, I also want to highlight something. These chromosomes that I've included here on the whiteboard and these chromosomes that I am drawing now, it's 6 and 9, is the same thing. How is it the same? If we go back to what I've said, I said you have chromosomes that are unreplicated and chromosomes that are replicated. So these two chromosomes, ladies and gentlemen, are also referred to as homologous pair of chromosomes. But now what's different between these two and these two? These two chromosomes are unreplicated and they're made up of one chromatid. So this will be chromosome 1 and chromosome 2. These chromosomes over here are replicated, meaning they are made up of two chromatids. So what I'm saying is this and this, same business. The only difference is unreplicated and others are replicated. All right. I'm going to do something. Let's see if I can erase a little bit or maybe use the space. Okay. My chromosome shapes are just too hectic. Okay. All right. Please do let me know. We are over here. How many chromosomes can you see? Just chromosomes. How many chromosomes? Anyone? We are over here.
I see 5.
5 is correct. 5 is correct. There's 5 chromosomes. There's 1, 2, 3, 4, 5. The only difference is that these ones are unreplicated. I think we're happy with chromosomes and number 46 and all of that. Let us do mitosis. I'm happy to take questions while I erase. So if you have questions, please do unmute and ask while I erase. It's probably going to take me just 5 seconds. I'm happy to answer any question, guys. Okay, there's nothing. All right. So just give me a little bit of time so that I can go through mitosis for you. I'm just erasing. Let's all mute. Okay. Done. Okay, please do mute. If you had unmuted because you were answering, please do mute. Okay, so now we are going to do mitosis. Now, if we remind ourselves, we said mitosis is a type of cell division that is going to take place in order for one cell to divide to form 2 cells. You need to remember this. In mitosis, we are having one cell that divides to form 2 cells. And these 2 cells are going to be identical to each other. We're going to find out how exactly do we start by having 4 chromosomes. We are not going to use 46 chromosomes because it would take up a lot of space if we were to do that. We are just going to use 4 chromosomes. Now, content says that if a parent cell has 4 chromosomes, the daughter cells should also have 4 chromosomes. So what we are having here at the end is 2 cells that are identical to each other. Now, mitosis itself is a process. So if we speak about a process, that means the stages or phases that are involved here. Now, if you want to bake a cake, that is a process. Now, there's certain stages that you need to follow. Mix the flour with water and eggs and milk. I think that's what people do. And then you do something else. Wait for the flour to rise or whatever. So now, mitosis as a process will take place in 4 stages or 4 phases. So you need to have a way of remembering the phases. So we follow this code of writing P-MET. P-MET. Now, these are first letters of the names of stages or names of phases here. I can include another one, but it's no longer regarded as a phase in mitosis. It's regarded as a phase in the cell cycle. If you remember it, interphase. When we did DNA replication, we spoke about DNA replication taking place in the interphase. So that is the first stage. So in this stage, this is where DNA replication will take place. The next stage that we are going to speak about. So if I were to draw cells, let me figure this out. How would I draw cells here? Okay, I can see what it does. Okay, so if I were to draw cells here, I will start with interphase. Now, what we will see in interphase is just the DNA of the cell replicating to make a copy of itself. That's the only thing that happens in interphase. Now, when this happens, we are also going to get the chromosomes because this network will then condense, become closely packed together and form these chromosomes. Now, that means in terms of interphase, we are done. Please make a note. I'm not able to make all the notes on my slides. In interphase, this is where DNA replication takes place. I've written I to indicate that is for interphase. Now, just after that, the chromosomes are going to be visible, and they will be visible in their replicated form. So that means if we had 4 unreplicated chromosomes, they are going to replicate to then become 4 replicated chromosomes. We are done with interphase. That's all that happens. Then the next phase is prophase. So the next phase over here will be prophase. So we're like, oh, yay, we are done with making a copy of DNA. The chromosomes have replicated, or DNA has replicated. Now, when we look at prophase, I'm going to mute this, I think 2 people or a person that has not muted so I can hear sound. If it's for a question, please do ask. If it's not for a question, please do mute. If it's for a question, I'm happy to answer that question. So now I have to go back here and try and mute everyone. Okay. So the next phase is prophase. Now, in this phase, this is where we start to see the chromosomes. Please note, in mitosis, the chromosomes are not in pairs. So we are not going to speak about homologous chromosome pairs anyway here. So these will be just the 46 chromosomes. Now, what we see in prophase, I'm going to write P there, will be the chromosomes. Now, these are the chromosomes. This is chromosome 1, and I said I'm going to use an example of 4 chromosomes. Chromosome 2, chromosome 3, and chromosome 4. But there was something that I was supposed to do here. Just give me a second. Let me see if it's going to be able to remove this. Okay, no, it's not removing it. That's fine. All right, we are having 4 chromosomes, but I want us to remind ourselves, where is all of this taking place? It's taking place in the nucleus. So in prophase, 1, we are able to see the chromosomes. 2, this is taking place in the nucleus, but when the process takes place, the nuclear membrane will start to disappear. So I'm drawing these dashed lines to represent that the nuclear membrane will start to disappear. So if you were asked to label, this would be your cell membrane. Please do label it on your side. And this will be your nuclear membrane. Remember, you find the nucleus in a cell. So if I were to draw it here, I don't want to finish up the space. This would be the cell membrane. And then this would be the nucleus made up of the nuclear membrane. And then this would be your genetic material. Now, what I've done on this side, it's the same thing. The only difference is the shape of the membrane. So this is the cell membrane. Please do write it. And this is the nuclear membrane. Now, in each phase, you need to be able to draw the phase. You need to be able to analyze the phase that you are given also and describe what is happening in that phase. So far in prophase, we are able to say the chromosomes are visible. That's point number 1. Point number 2, the nuclear membrane starts to disappear. That is the second point. Now, the third point, you can speak about the nucleolus. Remember when we did the nucleus and I said that's grade 9, grade 10 work. They're not going to test us about drawing the nucleus, but we need to know the information about the nucleus. So if you remember, the nucleolus was this big dot that we found inside the nucleus itself. So it will also start to disappear. And we are going to represent the nucleolus disappearing by writing dots inside the nuclear membrane. This will represent the nucleolus disappearing. That's not all. Another thing that will happen here in prophase, and all of these things you need to know how to describe. So here I spoke about the cell membrane. Here, sorry, see here I said it's the cell membrane, but then the key thing is you can see the chromosomes. And this point over here, we said the nuclear membrane starts to disappear. And this one is the nucleolus that also starts to disappear. What will also happen in prophase, there will be structures that are going to be formed. So on opposite poles of a cell, so north and south poles of a cell, we'll have a structure that is called a centriole, or you can call it a centrosome. Yeah, I think they take both. So this one I will need to label. So this is the centrosome or centriole, which is centriole. So even if you say centriole, you're going to get a mark. Now, what we then have here, we will have the centriole. You'll probably find it here in the middle. We say it starts to move to opposite poles. And as the centriole moves to opposite poles, these are two little structures that will move to opposite poles. As they move to opposite poles, they start forming what we call spindle fibers. I'm going to try and use a different color. So they start forming what we call spindle fibers, these small fibers that you're going to find here. And they are called spindle fibers. In other textbooks, they will say spindle threads. That will still be correct. Spindle. Come on. Spindle is Spindle. Spindle fibers. Okay. Fiber. Even if your fiber is the BRE, they take it, or the BER, it doesn't matter, they take it. This is a D. Now, let's understand this concept. What is happening here? These centriole move to opposite poles. As they move to opposite poles, they form these spindle fibers between them. So that means the top one, the top centriole and the bottom centriole are holding the spindle fibers together because they are forming as they move to opposite poles. You are done with prophase. Now, interphase, we said DNA replication. And prophase, we said the nuclear membrane starts to disappear. The nucleolus starts to disappear. Chromosomes are visible. And the centriole moves to opposite poles to form spindle fibers, formats. And then the next phase is metaphase. M for middle. Metaphase. Someone's mic is unmuted. Please do mute it unless you have a question for me. Then I'm happy to stop and listen to the question. Remind yourself, this M is for middle. Make a note. Now, these chromosomes in prophase are floating in the nucleoplasm because this whole process is taking place inside a nucleus. Then, when it gets to metaphase, that's the third stage here. In other books, it will say it's the second, and the reason for that, other books will not consider interphase as a phase of mitosis. They will consider it as a phase of a cell cycle. It shouldn't confuse you. It's 6 and 9. Now, in metaphase, these chromosomes are going to align themselves in the M middle of the equator. So now, when we look at metaphase, remember, this will be the cell membrane. And then we're not going to have the nuclear membrane because it has disappeared. Remember, we said in prophase, it was starting to disappear. Now, what we'll have here is these chromosomes, and we are going to become consistent with the number. We are having 4 chromosomes here. Now, that means these 4 chromosomes are going to align themselves in the middle of the equator. So I'm having 3 and 4 in a single line. This is important. They don't align themselves in pairs. So this is what I'm saying. In pairs would be if there is a chromosome here and another chromosome here, and then a chromosome here, and then another chromosome here. This would be chromosomes aligned in pairs. It is just one single line of the chromosomes, M for middle. But now, what's holding these chromosomes in the middle of the equator? Remember those spindle fibers that we formed in prophase? Remember we said the centrosomal centrioline moves to opposite poles, and once it does that, the spindle fibers that are going to hold, or the spindle fibers that will be formed, those spindle fibers that will be formed, there's a centrioline, there's another centrioline. They are going to hold the chromosomes in position. So that's what you're going to have in mitosis. In mitosis, metaphase, the chromosomes are in the middle of the equator, and we say they are there singly. Please make a note. They are there singly in the middle of the equator, not in pairs. If you say anything about a pair, you are going to get it wrong. Please note, we are still calling them chromosomes. And then,
Sorry,
yes,
ma'am.
yes.
Can you just repeat what you just said?
Singly. The chromosomes arrange themselves singly in the middle of the equator. And we're able to see this. Okay, so that's the key important point for metaphase. In prophase, we had 4 points. In interphase, we had 1 point. Now, please make a note, and this is a common mistake. The chromosomes are aligned in the middle of the equator. In other years, they penalize you for saying in the middle of the cell. So as much as me and you can see, they are in the middle of the cell, but they don't want you to say that. They want you to say middle of the equator, and they arrange themselves singly. They are not in pairs. We are then going to move. We are then going to move to another phase, which is now anaphase. That's the next one. We normally say this phase starts with A. Remember, A for apart. Think of that, make a note. A for apart. The name of the phase is anaphase. I'm trying to make...
Sorry,
Yes,
ma'am.
yes.
Before you move forward, I just want to ask if I see on my study guide it written as a chromosome in single row on equator. Is that correct?
Yeah. So the word single, equator, chromosome, those 3 words are important.
Okay, thank you.
All right. Why is it doing this? Okay, I'm going to find space somewhere here to speak about anaphase. Remember we said anaphase, A for apart. Now, this anaphase comes just right after metaphase. Remember in metaphase we said these chromosomes align singly in the middle of the equator. Now, these threads, spindle fibers or spindle threads, are going to contract. And remember, these threads are holding onto the chromosomes. They are attached onto the centromere. So if this is a chromosome and this is a centromere, the spindle fiber is attached to the chromosome through the centromere. Now, when they contract, they will contract and pull chromatids to opposite pole. Terminology is changing. So when they contract, the centromere is going to split. Whatever that is keeping the chromosomes together is going to split. And when that happens, the chromatids are no longer together in order for us to call them a chromosome. So they will be separated. So what are the words that we are going to use here? We can say chromatids are moving to opposite poles. We can say daughter chromosomes are moving to opposite poles. We can even call them unreplicated chromosomes, because remember, the main reason we are calling them a chromosome is because they are replicated. There's two of them, or there's two chromatids making up a chromosome. So the terminology changes here. So how will the diagram look like? Ooh, okay. So we need to show that the chromosomes are being pulled to opposite poles. So they will have a V shape. So if you look at these chromosomes over here, we are going to have these ones that I'm highlighting over here. These chromosomes are going to be pulled upwards. And then these ones that you have at the bottom are going to be pulled downwards to the opposite side of the equator. So let us show that in our diagram. So this will be the centromere, and the centromere has the spindle fibers that are attached to our daughter chromosomes. So we are going to show this V shape to show that actually this poor daughter chromosome has now been pulled, hence the V shape. There it is. Well, it looks like a heart, but it's actually a V shape. Anyways, it's Valentine's week, so I guess that's fine. But yeah, those are daughter chromosomes. Please note. Now remember, I only pulled up the ones that were on top because the centromere split and then the chromosomes moved apart. Hence A for apart. There is a separation that is happening here. So then we're not done. We need to include the other chromosomes. There's one, there's two. Okay. I'm trying to get space. Okay. Yeah. Then there's the third one. And there is the fourth one. Ta-da! It's easy for us to see the differences between the few phases that I've already drawn. In prophase, the chromosomes were just floating in the nucleoplasm. There was a line there showing us, a dotted line or dash line showing us that the nuclear membrane was disappearing, nucleolus was disappearing, spindle fibers were being formed. In metaphase, we saw that the chromosomes were aligned singly in the middle of the equator. Then in anaphase, the spindle fibers contracted. The reason we are having chromosomes looking like that, the spindle fiber contracted and the centromere split, and then the daughter, keyword, daughter chromosomes or unreplicated chromosomes were pulled, listen to me, opposite poles. If you say opposite sides of the cell, wrong. They are pulled to opposite poles. Okay. Then we go to the last phase, which is telophase. Let me see what happens when I click here.
Ma'am?
Okay. Is there a question? I'm hearing sound.
Yes, ma'am. If I said the centrosome pull towards the pole, is that correct?
I didn't hear centrosome does what?
Pull towards the pole.
Okay, let's remind ourselves, what is a centrosome? Let's remind ourselves, what is a centrosome? So a centrosome in this case is not pulled towards the poles. What is being pulled are these daughter chromosomes. These daughter chromosomes here. I'm not sure if I can use my highlights to show that the chromosomes are the ones that are pulled, not anything else. Yes, I'm highlighting them there. I'm not sure if you can see. This is what is being pulled to opposite pole, not the centrosome. The centrosome are at the opposite poles holding the spindle fibers. So we're not pulling them at all. Okay? Yes. So these structures are the ones that are being pulled. Hence, the change in shape. They are having this V shape now. So terminology will be a problem here. I am telling you, it happens every year. So it will need you to practice and go over your notes, because there's these three words that I've mentioned now. They sound the same, they look the same. Centromere, centralized, centrosome.
Yes, ma'am.
Centromere,
I think I was a little bit out because of network. I didn't hear you maybe when you were explaining some of the things. Sorry for that. I just wanted to clarify if is that correct, whatever I asked. So
oh,
okay,
okay.
thank you.
All right, thank you so much. So it is the daughter chromosomes that are being pulled to opposite poles. Okay, this is a mess, but let's go to the last phase, which is telophase. I'm going to draw it here and I'm going to represent it with the T. So if I go back to my initial notes, this was telophase. Telo, it's T-E-L-O, then phase, P-H-A-S-E. Okay, telophase is the last phase of mitosis. What will happen here, think of it. If this is a whole cell, I'm not sure it's coming out like that. And then now you are having these chromosomes that are pulled on one side of the cell, and other chromosomes are pulled to the other side of the cell. Now, it will get to a point whereby the cytoplasm of the cell will start to divide. So you have chromosomes here. We can even bring it back to the anaphase for you to understand. So if we have chromosomes over here, I'm taking it back to anaphase. So you have chromosomes this side, moving towards the upper side of the pole, and then you have chromosomes here at the bottom moving to the opposite side. That jelly-like substance that fills up the cell will then divide, but that is influenced by the cell membrane itself forming what we call a cleavage. So it will move inwards. It will form this type of a shape to form a cleavage. Once it forms this cleavage, I'm not sure if you need to remember this word. Yes, I think in grade 12 you do need to remember that. Once it forms this cleavage, the cytoplasm that was once in one cell is going to divide, and then the cell membrane encloses. We then end up having two cells. So when the cell membrane encloses, it is going to make sure that this part is closed. You then end up having two cells. What cells are we looking at here? So these chromosomes that moved upwards will form one cell, and these chromosomes that move downwards, the opposite direction of the equator, will then form the next cell. So what do we end up having here? This will be one cell, and this will be another cell. And in this cell, let's look at how many chromosomes we will have. 1, 2, 3, 4. We draw them. 1, come on. Okay. 2, 3, 4. Same with this cell. 1, 2, 3, 4. Let's look at it. You can unmute because we're almost at the end now of mitosis. Excuse me, of mitosis. So if we were to come back here on this diagram of our prophase, how many chromosomes did we start with? You can unmute and answer. How many chromosomes do we have in our parent cell in prophase? We call it a parent cell because that's the one that is going to divide. Okay, we're not moving up until I get an answer. 4 chromosomes.
4.
Thank you.
Yes.
How many cells did we end up with in telophase? How many cells?
2.
2 cells. How many chromosomes can we see in each cell? 4
4, ma'am.
Okay, what's different Let me see if I can use a different color or a pointer here. Okay, what's different between, we are saying we started with 4 chromosomes in the parent cell. And at the end of telophase, we are having 4 chromosomes in each cell. But something looks different here. What's different between the chromosomes that we find in prophase and the chromosomes that we find in telophase? Anyone can tell me. Unless I have to call out names. Okay, Slindo, what's different here? Slindo, what's different between the chromosomes that we found in prophase and the chromosomes that we found in telophase?
Can I answer?
Okay, that's fine.
The different is that on the last one, the last one, the last one, it's replicated.
So the last one you mean telophase over here?
Yeah, it's replicated. And
okay.
No, this one is unreplicated.
Which one? In life sciences, we don't know this one. Give us the name.
On the, okay, let me find the name. On the telophase.
Yes.
On the telophase. It's unreplicated.
Correct. Then on the prophase,
it's replicated.
Okay. As much as I'm saying it's correct, if I was marking you, I was going to penalize you. You are saying on telophase, it is replicated. What is replicated?
DNA.
We don't see DNA here. We don't see DNA. We are seeing chromosomes.
Okay, so sorry. Chromosomes are replicated.
In telophase.
Yes.
Or in prophase.
Or in telophase. In telophase are unreplicated.
Okay. So it's important that I do this because that's how we mark.
In telophase are unreplicated. In
prophase, they are replicated. So when we answer,
yes,
ladies and gentlemen, we need to answer in full. And that is why other people answer life sciences and say, yay, and then the marking comes back, the marks are low. We are very strict in life sciences. So I'm happy that we are able to do these interactions so that we can pick up those mistakes. So once you are asked to compare, you tell us what you are comparing, not it is, they are. We don't use that language. So you would say in prophase, the chromosomes are replicated, and in telophase, the chromosomes are unreplicated. So I think this should be fine in terms of mitosis. What we need to take away from this is the phases and that chromosome number is maintained. Whatever number that we start with in the parent cell will be the same number that we end up with in each of the 2 cells. The number of cells that are produced, 2, and the chromosomes are identical in terms of their DNA, and the chromosome number will be the same. Now, in terms of mitosis, what is the importance of mitosis? All of this is in the notes. Mitosis is needed for cell growth, 1. So when cells grow, they actually need to divide and grow. And that's the reason we are this big today. We were once very small babies, 1 year, 2-year-old. So the cells divided and then we managed to grow. Mitosis can also be regarded as a process that allows reproduction, asexual reproduction. I think for that, just take asexual reproduction or reproduction. without me going into details. Mitosis is also needed to repair cells. So if your skin or whatever, like were to get burnt on your skin, so after some time you will see that, oh, okay, that burnt part or that open part of your skin will eventually be closed up. And the reason for that will be mitosis. These cells in your skin will divide and form more cells that are the same in order to close up that area. Those are the importance of mitosis: growth, reproduction, repairing damaged cells. We close the chapter for mitosis. We are then going to speak about meiosis. Okay, I'm happy to take questions as I erase. I'm going to try and erase all of this. I'm happy to take questions while I do that. Erase entire stroke. Okay.
Ma'am, can we have key points on telophase before we move to my meiosis?
Okay, for telophase, two daughter cells are formed, or two identical. The word identical needs to be there. Sorry about that. Two identical daughter cells are formed.
Okay. Two
Yeah.
identical.
Yes. They will have the same chromosome number. So if we're having 4 cells in cell A, we'll also have 4 cells in cell B. If we are speaking about mitosis taking place in our body cells as human beings, remember we are having 46 chromosomes. So that means the parent cell will have 46 chromosomes and both the daughter cells will have 46 chromosomes. Okay, that is important to remember. All right, I'm trying to figure out how I'm going to do this. Let's just allow the pen to come back and then we cover meiosis. Anyone for 30 seconds who can tell me what they know about meiosis? Anyone? Now, I don't want only one person to be speaking in the tutorials. There's quite a few of us. So let's get people to speak, please, so that we can all learn. I need information about meiosis. It's not the first time other people come across meiosis. So I'm happy to listen up. There is an SK. I can't really see fully. Samuel, are you the one giving me the answer?
Hey, no, not now, man.
Okay, not now. Okay. Anyone, guys? We're not moving up until someone tells me about meiosis. We're not moving. We're not moving. Because my screen is still obviously trying to erase everything. So anything about meiosis, whatever you know about meiosis, whatever you know.
Okay, so I have a point about meiosis.
Thank you.
So with meiosis is a type of cell division that works with sex cells. Yeah, just to, so we know these are the ovaries, the testes, and also in it works in animals as well as in plants. In plants we have the anthers and the ovarians, the females.
That is all correct. That is all correct. Thank you so much for that. So something to note with mitosis, I think I forgot it and I'm remembering it now because of Label's answer. So mitosis takes place in body cells. So your skin cells or any of literally cells that make up your different organs, except your sex organs, right? Which is your testes and your ovaries. We speak about autosomes and gonosomes, but we'll do that in genetics. So let's get through it. I think my skin, wow, skin, because I was speaking about skin cells. My screen is clear now. Okay, one thing we need to note, when we look at meiosis, it is a bit different to mitosis because one, in mitosis, we said we are forming 2 cells. In meiosis, we are going to form 4 cells. So you need to be able to differentiate between the two. In mitosis, we formed 2 cells, and those cells were identical. In meiosis, we are going to form 4 cells, and these 4 cells will be non-identical. We say they are genetically different. That is the key word. Now, when we look at mitosis, mitosis will take place in your body cells, like your skin cells and all of that. Meiosis, on the other side, takes place in the gonads to form sex cells. So if we speak about the gonads in males, that will be the testes. And the significance of this is to have the chromosome number. In females, it will take place in the ovaries to produce egg cells. So in males, we are producing sperm cells. In females, we are producing egg cells. And that will be due to meiosis. Now, I've highlighted a few differences. Mitosis, we produce 2 cells. Meiosis, we produce 4 cells. In mitosis, it will take place in the body cells. In meiosis, it will take place in the gonads to produce sex cells. Now, in mitosis, we only had 1 division, whereby the cell underwent different phases to form the 2 cells, and that was it. So that is known as 1 division. It divided once. It was 1 cell that divided once, and we're happy. In mitosis, in meiosis, we are going to have 2 divisions. We are going to have a cell that will divide to form 2 cells. And that won't be the end of it. Remember, this was the end for mitosis. So the parent cell will divide to form 2 cells, and that's not the end. These 2 cells are going to further divide to form 2 other cells. And we say the cells divide twice. There is a second meiotic division. So there's 2 divisions. This will be regarded as division 1, and this will be regarded as division 2. In mitosis, the cell divided once. Now let's look at the chromosome number. In mitosis, we said the chromosome number is maintained. If I were to write it here, but it's going to take me time to erase again. If we had a cell with 46 chromosomes in mitosis, each cell at the end of mitosis, in this case, will also have 46 chromosomes. This cell will also have 46 chromosomes. The chromosome number is maintained. Now that is different in meiosis. This is showing you guys meiosis. The pen is doing its things, but it's meiosis, MEIOSIS. The chromosome number in meiosis is going to be halved. If the parent cell has 46 chromosomes, each of the daughter cells at the end of meiosis will have half the number of chromosomes. And in this case, it will be 23 chromosomes. So that means this cell will have 23 chromosomes. 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 The maths is not mathsing here. So if, in another example of meiosis, we had a parent cell that had 8 chromosomes, this cell will undergo 2 meiotic divisions. This will be the first meiotic division. And then these cells will continue and go through another division. And then the chromosome number for each cell will then be half the chromosome number of the parent cell. And the half of 8 in this case is 4. So that means this cell will have 4 chromosomes. This cell will have 4. Same applies with this one. Same applies with that one. Now what's different here? Mitosis chromosome number was maintained. In meiosis, chromosome number halved. Let's look at it now. Remember in meiosis, I spoke about 2 divisions. Now, when it comes to the names of the phases, we are going to indicate the division of that particular phase. So the names of the phases for mitosis and meiosis are the same. In mitosis, we are also going to speak about a prophase, a metaphase, an anaphase, and a telophase. The only difference here is we would need to indicate the name of or the number of that division. What do I mean? I'm going to separate this so that I can at least compare what is happening here. Okay. Where is this one from now? Okay, I'm going to erase. Please do be patient with me, guys. I think also the recording makes everything to be slower because now Teams is using a lot of things. Okay, so this will be the first division, the first meiotic division. The first stage or phase will be prophase. Remember, prophase was also the first one. And I said, remember, interphase can be ignored because that one happens just in the cell cycle before cell division. We follow the same format, which is PMath. I think when I was in high school, the teacher said, think of passing maths. You pass maths. It will be PMath. You write it down so that you can remember the order of the phases. So the first phase will be prophase. But since we are speaking about meiosis, and we are saying we first look at meiotic division 1. So this is when the cell divides to form 2 cells. That is meiotic division 1. If you write prophase, that is wrong. You have to tell us the division number. So this will be prophase 1. If you don't write the number, wrong. Now, what happens in prophase 1? Now, the chromosomes are going to be visible, but listen to the terminology that we use here. It's not just chromosomes. The chromosomes are going to be in pairs. And for the sake of our example, I cannot draw 46 chromosomes. So I'm just going to draw 4 chromosomes. So this would be the cell membrane. And the same thing that we spoke about 2 things disappearing in mitosis prophase. Those very same things are going to also disappear in prophase 1. So in prophase 1, we will have the nuclear membrane disappearing. That's what you would need to note. And then the chromosomes are going to be visible. But in prophase for mitosis, there were just chromosomes visible. But in prophase 1, it is now the pair of chromosomes. So you will have this chromosome, and they will lie next to each other for us to see that they are a pair. And then this chromosome with its pair. It would actually look nice if I was using 2 different colors here. Because remember, when we speak about a homologous pair of chromosome, that means one is from the mother, one is from the father. Let me actually try and do that. It will make a huge difference. Okay, if I can remove this one. Oh man, it's removing the whole thing. Okay, so this is 1 chromosome. So the red ones, we will say these are the ones from the mother, and the purple ones are the ones from the father. So I'm going to ask a question. How many pairs of chromosomes do we see on our screen? How many pairs of chromosomes?
4.
How many pairs of chromosomes do we see?
2. 2.
4.
Two.
Four.
Four.
It's two. Two.
Sure.
It's two because two is blue and three is red. So it's two.
Okay, I'm gonna ask again because there's two answers. One is correct, one is not correct. The question is, how many pairs of chromosomes do you see?
Two.
Two.
No, we see two.
It is two
Two.
pairs of chromosomes.
The question is confusing.
But the question is not confusing. I repeated it 3 times. How many pairs? Now remember, a pair will be made up of 2 chromosomes. And in this case, the chromosomes have replicated. So this will form one pair and this will form another pair. So there's 2 pairs of chromosomes. Now I'm going to ask again, how many chromosomes do we see in that cell?
Four.
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 4, correct, 4 chromosomes. How many chromatids do we see in that cell?
8.
8.
8.
Yeah, you might as well go on and write the exam then. If you're able to tell this, then you should be able to get everything right. Okay, there's something that I forgot to include here. It was that structure that disappears, which is the nucleolus. So you can also see it, it's on the diagram there. It is disappearing. So in terms of the nucleolus disappearing and the nuclear membrane disappearing, that also happened in mitosis prophase. Now, what is different between, this is going to take time if I compare each phase, but what's different between prophase 1 and prophase of mitosis is that the chromosomes in prophase 1 are in pairs, and the chromosomes in prophase of mitosis were not in pairs. They were just randomly in there. Now, the same thing about spindle fibers will also take place here. So the centrioli will move to opposite poles and start forming the spindle fibers. It's normally indicated as that you just see them on the side because it shows that they are beginning to form. Now, there is a very important process that will take place in prophase 1, which is called crossing over. The process that will take place in prophase 1 that is very important is called crossing over. Now the pen is lagging again. Okay, there it's coming. Crossing over. Now this is very important. Now the process of crossing over takes place between a pair of homologous chromosomes. Now, as you can see, these homologous chromosomes over here, they are close to each other. So it's a chromosome from the mother and chromosome from the father. Now, these chromosomes are going to be very close to each other. I am going to start by drawing the chromosomes. Remember I said the red ones are from the mother, and then the purple ones are from the father. So this makes up one pair of homologous chromosomes. So they will start by coming very close to each other. Then the chromatids, check the terminology. The chromatids that are next to each other. So this chromatid from the female chromosome and this chromatid from the male chromosome are going to cross over. What does that mean? So if we're to go back, now they first come in close contact, and then this is the red one. So the chromatid that is next to a chromatid from the father will then cross over. So this would be the other chromatid, and this one will cross over. I hope you can see what is happening here. So these chromatids have crossed over. We say these are chromatids from the paternal and maternal, meaning from the mother and father or father and mother. So these two chromatids cross over here. Now, I don't want to confuse you, but we say these non-sister chromatids. We call them sister chromatids when they make up one chromosome. So this will be a sister chromatid, this will be a sister chromatid. This will be a sister chromatid, this will be a sister chromatid. Those are chromatids that are joined together by a centromere. These ones are non-sister chromatids because this is a chromatid from a pair of the mother, and this is a chromatid from the father's pair. They are non-sister, but you don't, I know the terminology will be hectic, but you can remember it. You can remember it for exam. So then they cross over. Now, this point of crossover, where these chromosomes then cross over, is called chiasma or chiasmata. So that point over there, where both chromatids have crossed each other, is called chiasma or chiasmata. One is singular, one is plural. Now, what really happens when they then cross, when these chromatids cross each other? Genetic material is going to be exchanged. Now, the DNA that was found in this chromatid will now be found in the other chromatid. So they basically exchange genetic material. That's the significance of this process. So we then end up having something like this. So this was the chromatid, the first one that did not undergo crossing over. It was this one that underwent crossing over. But because of the genetic material that has now been exchanged, it will have a purple over here to represent that it got the genetic material from the father due to this exchange of genes. And then the chromosome from the father, we said it was purple. There's the chromosome from the father with its two chromatids. But now what do we have here? At the bottom here, due to this exchange of genetic material, it will then have, we are just representing it by color, it will then have the genetic material from the mother that is crossing over. They will 100% ask you about this process. Where does it take place? In prophase 1. How does it take place? The pair of homologous chromosomes will lie next to each other. Non-sister chromatids will then overlap. At this point over here in chiasma, we say they overlap at a point called chiasma, and then exchange, overlap and cross over to exchange genetic material. At the end, you will have a homologous pair of chromosomes, but now the chromatid from the mother has genetic material from the father. The chromatid from the father has genetic material from the mother. Now, this is the reason you have the same mother and father as your sibling, but you don't necessarily look the same. You don't look like twins. You look very different. It's because of this process. Because now the genetic material has now been exchanged. It's not the main reason you look different. There's other reasons. Crossing over takes place in prophase 1. We are then going to continue. I'm going to erase because I want to be able to write down what happens in the next phase. So that happens in prophase 1. Now, the next phase, if we remember our P met, so that means the next phase will be met, which is metaphase. M for metaphase, M for middle. So that means it's going to be easy from now on. M for middle means that the chromosomes are going to align themselves in the middle of the equator. How? They will align themselves in pairs. In prophase of mitosis, we had chromosomes just being random. In metaphase of mitosis, they arrange themselves singly in the middle of the equator. We need to remember that. So what we have over here, I'm going to separate. Come on, come on, come on. I'm going to separate this and then write metaphase. Now with metaphase, what are we having? We are having these chromosomes in pairs in the middle of the equator. This is metaphase. Metaphase. Okay, this is the cell membrane, and then I'm going to show the chromosomes. So this will be the one chromosome. Come on. This would be the one chromosome, and this would be the other chromosome. Now, this happens randomly. I know I'm doing it here as if it goes in a particular order, but this happens randomly. So it can be any pair arranged on any side, upside down, or whatever. We say that chromosomes arrange themselves randomly in pairs in the middle of the equator. There it is, and there it is. So how do we analyze this? This is a pair, homologous chromosome pair in the middle of the equator. But what's holding these chromosomes steady in the middle of the equator is the spindle fibers. These are the spindle fibers. The spindle fibers, remember, are formed by the centrosome or centrioli. Now, if we were to be consistent here, I would be consistent with the crossing over and the exchange of genetic material here in my chromosomes. Now, the red ones have a little bit of a purple drawing there because of the exchange of genetic material. And the purple ones, I will draw a little bit of red to indicate the exchange of genetic material. So that is metaphase. I'm actually wrong. I should indicate 1 there. Metaphase 1. If you don't write the number, wrong. Now, how would you phrase this? Homologous pairs of chromosomes arrange themselves in the middle of the equator. Keywords: homologous chromosomes in pairs, middle, equator. You get a mark. Remember, in metaphase of mitosis, it was just single arrangement. Then we're done with metaphase. We go to anaphase 1. In anaphase 1, A4 apart. In anaphase 1, A4 apart. Now, I want us to look at what is happening here. Now, remember, in metaphase 1, these chromosomes are arranged randomly. As much as in my diagram, it looks like, oh, the chromosomes from the mother are going to be on top and the chromosomes from the father are going to be at the bottom. It's not like that. In this pair, it could be the mother's one and the father's one at the bottom. In this pair, it could be the father's one and the mother's one. In the other pair, it could be the mother and the father. If we're speaking about me and you, it's 23 pairs of chromosomes trying to arrange in the middle of the equator. Now, in anaphase, I want you to look at this because that's when terminology becomes important. So these chromosomes are in pairs. So this is pair number 1, my 2 fingers, and pair number 2, they are like this. So they are going to be pulled by the contracting spindle fibers. But I need someone to unmute and tell me what is being pulled to opposite poles. Are we pulling homologous chromosomes to opposite poles? Are we pulling daughter chromosomes to opposite poles? Are we pulling chromosomes to opposite poles? Are we pulling chromatids to opposite poles? What exactly are we pulling? I need the terminology here. I need you to tell me what exactly is being pulled to opposite poles by looking at the chromosomes that you have in your metaphase. Anyone who can help me? What are we going to pull to opposite poles here?
We are pulling chromosome.
Thank you. We are pulling chromosomes. Let's look here. Here they are in a pair. So we are going to separate them. We are going to ensure that this chromosome as a whole is pulled up and this chromosome here as a whole is pulled down. So what are we going to have at the end? This is one chromosome with two chromatids. And this is another chromosome with two chromatids. And then this one, same. The chromosome, full chromosome with two chromatids. Another chromosome with two chromatids. Look at the shape. They are being pulled to opposite poles. That is anaphase 1. The keyword chromosomes are being pulled to opposite poles. That's the keyword. If you don't want to use the word chromosomes, just mute, please. If you don't want to use the word chromosomes, you can say replicated chromosomes are being pulled to opposite poles. Please note, you cannot say chromatids are being pulled because here it's the whole chromosome. We are separating that pair. Now, I was supposed to be consistent here in terms of my color pattern. I was supposed to have the purple ones and then the red ones and the red chromatids that had a purple dot and the purple chromatids that had the red dot. I was actually supposed to be consistent here. But I guess we see what is happening. And then at the end of meiotic division 1, we will have telophase 1, PMET, in that order. So this will be telophase 1. Now, what will happen in telophase 1 is basically a continuation of anaphase 1 there. The cytoplasm is going to divide. And when the cytoplasm divides, let's look at it here. So the cytoplasm will divide in order to form two cells. Now, these chromosomes that will be pulled to the upper part of the pole are going to form one cell. And these chromosomes that were pulled downwards will form another cell. So what do we end up having here? We end up having two cells which are not identical. They are not identical because remember, we had an exchange of genetic material. All of those 23 pairs experienced crossing over. So the genetic material will not be the same. So we'll have this cell over here and this cell over here. Let's look at the number of chromosomes, whatever that is happening with chromosomes now. So what do we have here? We're having a full chromosome and another full chromosome. So there it is, one chromosome that is replicated and another chromosome that is replicated. There you go. Same applies in the other cell. This is one chromosome replicated and another chromosome replicated. Now, I want us to look at what we have here. Let's look at how many chromosomes we started with in prophase 1. And in prophase 1, remember, we had one cell. We had 4 chromosomes. And they were in pairs, meaning we had 2 pairs. How many chromosomes do we have in each cell at the end of telophase 1? We are having 2 chromosomes in each cell. And they are replicated. We started with 4 replicated chromosomes. We end up with 2 replicated chromosomes in each cell. So that means the chromosome number has been halved. So if I bring it back here, that means we started with 4, And then in each cell, we are having 2 chromosomes. That is the first division. Now we're not done. These 2 cells are going to continue to divide in meiotic division 2. So this cell is going to divide in order to form 2 cells, and this cell is also going to divide to form 2 cells. So I am going to show you these cells. I'm running out of time, but these are the 2 cells in meiotic division 2. These are the 2 cells, the same 2 cells that we had in telophase 1. They continue to divide. We then say that is meiotic division 2. Once they continue to divide, we then label that phase as prophase 2. We are starting over with the names of the phases. We label that as prophase 2. Why 2? Because we are saying it's the second division now. One cell divided to form 2 cells. Those 2 cells are going to continue to divide, but they start over in terms of the phases. Prophase 2. Now, there won't be crossing over taking place in prophase 2. Crossing over doesn't take place twice. It takes place only once. Now, how many chromosomes did we have in each cell in telophase 1? We had 2 replicated chromosomes. So there's your replicated chromosome. This is the first one, and this is the second one. This is the first one, and this is the second one. Now, let's remind ourselves of the key things that happened in prophase 1 outside of crossing over. The nuclear membrane started to disappear. It will be the same thing that takes place here. The nuclear membrane will disappear, and the nucleolus will also disappear. Nucleolus will also disappear. We are starting over. What happens to the spindle fibers? They will be formed. I'm going to draw them here. They will be formed, and the spindle fibers are going to be formed. We're done with prophase 2. Metaphase 2 is the next phase. Metaphase 2. Now, what happens in metaphase 2? Still, we are going to have an alignment of chromosomes randomly in the middle of the equator. But how are they going to arrange themselves in the middle of the equator? They will arrange themselves randomly, singly in the middle of the equator. The arrangement of which chromosome aligns itself after which one on which side is random. So whatever that I'm saying in prophase 2 and metaphase 2 and anaphase 2 and telophase 2 is the same thing that I mentioned in mitosis. So let's look. In metaphase 2, we are now having these chromosomes randomly arranging themselves. How many do we now have? We've got 2 chromosomes in each cell. So I don't want to confuse people. Let me draw 2 cells, because once I draw one cell, I know I'll be confusing people. Okay, come on. Come on, dude. Okay. Just give me a second. I'm trying to erase here. Come on. I'm scared it's gonna erase the whole thing. Okay, that's fine. Okay, you guys are too quiet. I'm not sure if he's still there. I hope he's still there. Okay, this would be a cell, and this would be another cell. Remember, we're working with 2 cells here. So in metaphase 2 for this cell, this is the chromosome in the middle of the equator, and this is another chromosome in the middle of the equator, held together by spindle fibers. Held together by spindle fibers. Same applies on the cell. This is a chromosome, and this is another chromosome, held together by spindle fibers, held together by spindle fibers. They should be perfectly aligned in the middle of the equator, but mine, they just don't want to align perfectly. And this is random. Now, I hope by now you are comparing your diagrams, your metaphase diagram 1 with your metaphase diagram 2. You are comparing the stages of the division 1 with the stages of division 2. Then when we have anaphase 2, so when we look at anaphase 2, we are going to have a separation here. But what separation are we having? If this is a chromosome, the centromere in the middle is going to split, and then what will be pulled to opposite poles is the daughter chromosomes. So if this is the cell and this is another cell, this is the same thing I did in mitosis. So this will have a daughter chromosome, a daughter chromosome. If you say a chromatid is pulled, we can still take that. Here it is pulled. Okay, my energy is depleting now. But yeah, let's just push. We're almost done. And this is another daughter chromosome, another daughter chromosome, or you can say unreplicated chromosome. Sorry. I hope you can see what is happening here. Then at the end, the cytoplasm is going to divide. When the cytoplasm divides, it will be at a point whereby our daughter chromosomes have reached the poles. So then when it divides, it is going to ensure that these chromosomes form part of one cell, and these daughter chromosomes here form part of another cell. Same applies this side. These chromosomes, daughter chromosomes, form part of a cell, and these chromosomes will form another cell. So that means at the end, we are having how many cells? This will be a cell. This will form a cell. These ones will be on their own cell, and these ones will be on their own cell. Now let's look at the chromosomes here. Let's draw them. So this cell over here, this will be one unreplicated chromosome and another unreplicated chromosome. Here, same business, unreplicated chromosome, unreplicated chromosome. Same business, unreplicated chromosome, unreplicated chromosome. Same business, unreplicated chromosome, unreplicated chromosome. We are reaching the end. I need you to unmute. Now this is your time. We are going to look at what is happening in each phase. Look at prophase 1, prophase 2. This would be anaphase 1, and this, anaphase 2, and this would be telophase 2. Now I'm going to ask questions so that we can really see what is happening here. And when you started this, you should also write it like this. You can write it horizontally or vertically in terms of your phase names. How many chromosomes did we have in prophase 1? Please tell me.
4.
4 chromosomes. How many chromosomes did we have in prophase 2 in each cell?
2.
2
True,
chromosomes.
ma'am.
So the chromosome number is halved. So the cells that undergo the second meiotic division already have half the number of chromosomes.
Okay.
This is consistent. Yes, is there a question?
No.
Okay, so this is consistent with the rest of the phases. Let's look at metaphase. In metaphase, we had homologous pairs of chromosomes in the middle of the equator, arranged randomly. So that means in total, we had 4 chromosomes in metaphase 1. But in metaphase 2, in each cell, we had 2 chromosomes, half the number of chromosomes. Same applies with anaphase. In anaphase 1, we had a total of 4 chromosomes, 2 chromosomes pulled to each pole. But when we look at anaphase 2, we actually had 1, 2, 3, 4. We actually had 2 chromosomes, and in this case, they are daughter chromosomes being pulled to opposite poles. Check here. In anaphase 1, it's a full chromosome that is being pulled. In anaphase 2, it's a daughter chromosome, or it's an unreplicated chromosome. Let's look at telophase. How many cells did we form in telophase 1? How many cells?
Two.
Two cells.
How many cells did we get at the end of telophase 2?
4, ma'am.
4 cells.
Thank you so much. In metaphase 1, it's homologous pairs of chromosomes that are arranged in the middle of the equator. In metaphase 2, it's chromosomes arranged singly. They are not in pairs. You can see them. In anaphase 1, it is the whole chromosome that is pulled to opposite poles. In anaphase 2, it is the daughter chromosome or unreplicated chromosome that is pulled. Now let's look at the chromosomes that we find between telophase 1 and telophase 2. Because in each cell for both phases, we are finding 2 chromosomes. But the only difference is that the chromosomes that we find in telophase 1 cells are replicated. But the chromosomes that we find in telophase 2 cells are unreplicated. So if we were to summarize this, I'm trying to get space. So we started with a parent cell. My brain is shutting down. And our parent cell had 4 chromosomes. Meiotic division 1 occurred. And when meiotic division 1 occurred, we had 2 cells at the end of telophase 1. Now, in each cell of telophase 1, we had 2 chromosomes. And then these 2 cells continued with cell division and produced 2 cells each. 2 cells each. There you go. There you go. There you go. And in all of these cells, at the end of meiotic division 2, we found 2 chromosomes in each. Now, this, when you look at this, it can look confusing. But if you come back to this diagram, it should not be confusing. We said the difference between the chromosomes that we find in telophase 2 and the chromosomes, in telophase 1 and the chromosomes that we find in telophase 2 is just that the chromosomes found in telophase 1 are replicated, but they're actually 2. Even if you come here, you can count, it's 2. And the ones that we find at the end of telophase 2, they are also 2. But the only difference is that here they are unreplicated, and over here they are replicated. Now that is all the juice about meiosis. There's obviously quite a lot that I still need to emphasize, especially terminology. But I'm happy to take questions just based on the lesson. I'm happy to take questions, but there's a lot to clarify and go over next week. It takes about 7 days to teach this, so I can't do it in 2 hours. But I think I've covered all the important parts. I'm seeing a few people. I'm not sure if you guys want to ask questions. Samuel, Slindo?
No.
Okay.
No, ma'am, sorry.
Okay, that's fine. I will send a homework activity on the group. Please do attempt that activity. The terminology is hectic here. The words sound the same. 1 minute it's a chromosome, the other it's a chromatid. After 2 minutes it's a daughter one. Another 3 minutes it's an unreplicated. This can be confusing. So it will need a little bit of practice. Like I'm saying, we normally take about 7 days to teach this. Because we start by teaching mitosis, which can take 2 lessons, and then meiosis, another 3 lessons, and then we speak about, we compare the similarities and the differences. But we will do that. So that means next week we are going to summarize everything and look at our slides so that all the content or even things that I forgot to mention, I make sure that I mention. All right, I'm happy to call it a night. I will send you the homework activity on the group. Can we say bye?
Bye,
Okay,
ma'am.
ma'am. Thank you very much. Bye.
Thank you.
Bye.
Bye, everyone.
Bye.
Bye.
Don't forget to send us the recording.
Okay, I hope it recorded everything. I think it did. Let me see.
Because at the first 15 to 20 minutes I was not here, so I just came after. So I just wanna even go back to what I didn't hear any. So